Hello, my name is Karl Schumacher, and this is my final honors thesis and bioengineering senior design project titled Ambulatory Monitoring of Biomarkers for Enhanced Recovery. I completed this project with help from my project partners, Oliver Holman and Ben Surdi, and we were advised by Dr. Uyung Kim and Dr. Mariam Mobed Miramati from the bioengineering department. The goal of this project was to develop a wearable biosensor medical device which is able to monitor the progression of chronic diseases like Alzheimer's and cancers through the continuous monitoring of reactive oxygen species in the body. We accomplished this by developing a three-component device consisting of a microneedle array, an electrochemical gel, and a printed circuit board. Our device works by specifically detecting hydrogen peroxide concentration, which is correlated with the progression of chronic diseases, as well as it is a systemic biomarker, meaning it can be found throughout the body. The first and bottommost layer of our device is the hollow microneedle array, which is composed of many small, short needles which are able to gain access to the interstitial fluid underneath the skin. Our needle arrays were fabricated using stereolithography 3D printing and were optimized to maximize the sharpness of the needle tips as well as the ability to resolve a small hollow bore for fluid transport. The needle sharpness was tested using static force puncture tests to find the force per needle required to successfully achieve 95% puncture rate in a skin simulant. Additionally, the needle bore geometry was tested for its ability to transport fluid using capillary action. Both of these test results informed our final needle design and the critical dimensions that were needed to ensure that they would reliably puncture and effectively deliver interstitial fluid containing the biomarker to the next components of the sensor. The next layer in the device is an electrochemical gel, which is a chitosan-coated alginate gel containing the enzyme catalase and single-walled carbon nanotubes. This reacts with the hydrogen peroxide in an ISF sample to produce an ion mediator. This proportional ion concentration can then be converted into an electrical signal through a process called amperometry, which occurs between a carbon and silver electrode array in the presence of a working voltage. We used many measurements of the amperometry current response at various concentrations to construct a regression model that correlates the electrical output to the hydrogen peroxide concentration in the sample. The final and top layer of the device is the amplifier PCB, which performs the amperometric measurements and takes the small hydrogen peroxide signal from the electrochemical gel and converts it into a readable voltage. The PCB contains the electrode array and a tunable gain parameter for the amperometry cell, while still being optimized for its size to be comfortably wearable. The final step of our involvement with this project was integrating a complete prototype containing all the layers, which allows a future group to begin fine-tuning the device's performance in a clinical application. In conclusion, we hope that this project will continue its development into a clinical or research role that is able to help patients living with chronic diseases. Specifically, we see it being able to truncate the cycle of disease that patients usually live within and prevent them from ever reaching a state where they're at risk of an acute event requiring a major intervention by continually providing them with personalized high-resolution data about their disease progression and treatment effectiveness. Finally, I would like to acknowledge the people and organizations that made this project possible. Our academic advisors, Dr. Kim and Dr. Mobed, the Santa Clara Engineering Department for the project funding, and Jai Wang at the SCU Maker Lab for his help with our microneedle fabrication. And thank you for listening.